the first question we have here is 1:1:1:1, alright, ratio shows. 4 options have been given: monohybrid cross, dihybrid cross, back cross and dihybrid test cross. Dear students, this ratio, alright, can be seen with respect to dihybrid test cross. Let me explain. If we consider the parental generation to be round yellow, alright, that is capital R, that is round yellow, okay, fine, wrinkled green, the genotypes for the same would be, alright, capital R, capital Y, small y. For this, the genotypes would be small r, small y, small y. Now, after the meiosis occurs, alright, the gametes would be formed from, the gametes would be formed rather. So that would be, for this it would be capital R, capital Y, and then capital R, small y. Then we have small r, capital Y, small r, small y. For this it would be small r, small y, right? First one, when it crosses with the small r, small y, we are going to get capital R, capital Y, small y. Then the second, when it crosses with the small r, small y, we are going to get capital R, small y, small y. Then the third one with small y, we are going to get small r, capital Y, small y. And lastly, the fourth one with small y, we are going to get small r, small y, small y. See, like this 4, alright, phenotypes would be obtained. The first one is round yellow, and the second one we have obtained here is round green. The third one we have obtained here is the wrinkled yellow, and the last one we have is wrinkled green. So phenotypic ratio we are going to get is 1:1:1:1. So in the case of dihybrid test cross, alright dear friends. So what is dihybrid test cross? A cross produced between the F1 hybrid with a double recessive plant generation or parent generation, sorry. So when we consider this, we are going to obtain the ratio 1:1:1:1. So go for option 4, that is your right answer. Then dear friends, the next question we have here is the genetic, defect adenosine deaminase deficiency may be permanently cured by, alright. So what is the options here? Enzyme replacement therapy, periodic infusion of genetically engineered lymphocytes having functional ADA complementary DNA, administering adenosine activators, or introducing bone marrow cells producing adenosine deaminase into cells at early embryonic stage. Dear friends, see here, to get rid of or to permanently cure the defect of adenosine deaminase, that is what we call it to as severe, alright, combined, alright, immunodeficiency, which can be in simple sense referred to as SCID, alright. To cure this severe combined immunodeficiency, alright, which is a life-threatening syndrome, alright, where recurrent infections can be seen, alright, where we can also find the recurrent diarrhea, alright. And dear friends, along with that, even dermatitis can also be one of the common symptoms associated with SCID. And failure to thrive, survival becomes difficult, because the complete immune system will break down, my dear friends. Here T lymphocytes will also not be sufficiently produced, because adenosine deaminase is very much essential for the production. Alright, now here let me tell you, my dear friends, SCID patients can be cured with, alright, the HLA matched sibling, alright, what we it is HLA matched sibling, alright, sibling donor, he should be HLA matched. And also, alright, this can be possible through bone marrow, alright, transplantation, alright. So definitely the fourth option would be correct here, alright. The only chances of permanent cure is what? Introducing bone marrow cells producing ADA. That means from the healthy person we have to take these cells which produce ADA, and has to be, alright, put in at the early embryonic stage itself into the, alright, the patient. Then we can say there is permanent cure. Chances are definitely there. So right, the more appropriate option we have to go for here is option D. Dear friends, the next question we have here is, if 3 to the power n is the theoretically possible number of, see 3 to the power of n, if it is theoretically possible number of genotypes when n is number of chromosome pairs with each carrying one pairs of heterozygous allele, the different types of genotypes by P plant is. Please understand, dear students, here first point to the note here, according to the question, if we observe possible number of genotypes, see different genotypes they are giving it as what, 3 to the power of n. So here possible number of genotypes would be, this is possibility, genotypes would be 3 to the power n, alright. And here n represents what? The n is representing number of chromosome pairs, alright, with each carrying one pair of heterozygous allele, alright, as mentioned in the question, this n, alright. Now dear friends, let me tell you that the number of homologous chromosome pair in P plant, homologous, alright, chromosome pair, alright, the number of chromosome pair in P plant is 7, alright my dear friends. So hence the possible number of genotype for P plant would be how much? 3 to the power of 7, alright. So that is how you have to go for, alright. Let me repeat, 3 to the power of n is number of possible genotypes, alright. So where n represents what? N represents here the number of chromosome pairs, alright, with each carrying one pair of heterozygous alleles, alright. If that is we consider, then in P plant homologous chromosome pair is 7. So it would be, alright, genotypes, possible genotypes in the P plant would be 3 to the power of 7, alright. So please go for option, alright, 4. Here in the power it should be 3 to the power 7, my dear friends. All these are 3 to the power of 7, this is 3 to the power of 14, 3 to the power of 10, and the second option is 3 to the power of 12. It should be in powers anyway. So the right answer is 3 to the power of 7. So please go for it. Then dear friends, next we have, if a colorblind woman marries with a normal man, the offspring will be, alright. Again it's an interesting question. Okay, let me tell you that first of all, color blindness, alright, it's a color blindness is a, alright, X-linked recessive disorder, alright. It is linked with the sex chromosome, alright. So we refer it to as what? X-linked recessive disorder. Only in recessive condition it affects. And dear friends, let me also tell you, it follows zigzag or crisscross inheritance, alright. That means to say that, alright, we are well familiar with it. The grandson inherited color blindness from his grandfather, alright. From the grandfather, grandson got the color blindness through his mother. That's what we call it as crisscross inheritance, my dear friends, alright. Anyway, here let me explain you what is the offspring will be. The colorblind marries colorblind woman marries a normal man, what will be the offspring is the question here. So let us solve this. See, option 1 is all are colorblind. Second option is all daughters normal and all sons will be colorblind. Third is all are normal. Fourth option is all daughters will be colorblind and all sons will be normal. Here dear students, say let us consider the parental generation to be, alright, the XC and the, alright, XY, alright. XY here is male, XC here it is indicating the, alright, colorblind woman. So now gametes from this would be here XC, here it would be XY. So these are the gametes, alright. Now what are the possibilities here? When see this combination, we will get XCX, alright, and then secondly we are going to get XC and Y, alright. Then thirdly we are going to get XCX, and fourthly we are going to get here XCY. Alright, see the first one here, it is carrier daughter, and XCY which is represented here is colorblind son, and XCX here it is carrier daughter again, and finally XCY what we have here is colorblind son. Dear friends, you can note down that the affected person would be son only, because the daughter will be always carrier. So among the given options we need to go for, as I said, all daughters will be color blind. Alright, all daughters will be here the right answer is option B, that is all daughters are normal, they will be generally carriers, alright, or normal, but the sons will be affected, all the sons will be color blind. So that is the condition here, my dear friends. So you have to go for option B. Alright, it follows the crisscross inheritance. Then dear friends, the next question we have here is, marriages between close relatives should be avoided because it includes more. Alright, 4 options have been given. The marriage between close relatives is called as consanguineous marriage, dear friends. Alright, so now why it should be avoided? The 4 options have been given. Recessive alleles, recessive alleles to come together, then mutations, multiple births, blood group abnormalities. Dear friends, here see, marriage between related individuals are risky, of course, and it is very much what I can say, the chances will increase for genetic disorders. Alright, so anyway, so let me explain you that this will be risky because see, the recessive alleles in the gene pool will accumulate. Alright, the recessive genes, whatever the recessive genes are, it will accumulate, alright, in the gene pool. Alright, they accumulate in the gene pool, my dear friends. This is the first point you need to know. If let us say the recessive allele, if it which reaches the female individual is heterozygous, female will be a carrier. Heterozygous condition, if it reaches, female will be a carrier. Alright, then gene gets transferred to father generation, definitely in the male child defective gene will be expressed. Alright, see, in heterozygous condition, if it is there, it will affect the female. Alright, and second point to be noted is that gene, if it gets transferred to further generations, alright, to whoever it is, alright, so what happens generally here, the male child, in the male child the defective gene may be expressed, my dear friends. Alright, so anyway, that's what the chances are there that the genetic disorders may be caused. So the right answer which you can go for here is what? Recessive alleles come together in the gene pool in when close relatives marry with each other, when consanguineous marriage occurs. Alright, so the answer is A, please go for it. So the next question we have here is, the 1900 AD is highly significant for Genesis due to the 4 options have been mentioned here, friends. Out of this we need to identify which was that one of the incidents which really changed the way we see today the genetics. Alright, genetics has developed today into modern genetics. Alright, so a lot of research have been involved with respect to genetics today, and this is all because 1900 AD there was a significant change around. Alright, let me tell you what happened here. See, firstly here the Mendelian rediscovery. Alright, Mendelian rediscovery here took place. The 3 scientists, the first one was Hugo de Rais, alright, then followed by one more scientist called Carl Correns, alright, then one more scientist Eric, alright, Chemark. Alright, these 3 scientists independently they rediscovered the Mendel's work. Alright, they rediscovered the Mendel's work, my dear friends. Alright, and this paved the way for more research in genetics, all right, and on Mendelian genetics as well, all right, my friends, and laid the foundation for, all right, the modern genetics. And that is why 1900 AD is highly significant for Genesis, all right, and it cannot ever be forgotten. Anyway, my dear friends, let us also know what are the other options given. Discovery of genes they have given. So there was one botanist scientist, all right, that is called as the, the name is here, Danish botanist called as, all right, Wilhelm Johansen, one of the scientists called Wilhelm Johansen, all right. He coined the term, all right, gene in the year 1909, all right. He took this word from, all right, the word called genetics. This genetics, that word was coined by William Bateson, all right, one of the well-known scientists at that time, all right, William Bateson. So from the genetics, he took that word and coined the term called genes, and later on, the genes, the study and the discovery took place. Furthermore, my dear friend, they have given principle of linkage. All right, again, William Bateson. All right, is very significant here to be remembered, my dear friends. All right, see here. Apart from that, let me also tell you, T.H. Morgan is called as the father of experimental genetics. He has worked a lot on linkage, my dear friends. Okay, here furthermore, if I want to tell you that, through the linkage, T.H. Morgan was able to define many important physical association of genes on a chromosome, all right. Anyway, for then we also have chromosomal theory of inheritance, Sutton and Boveri, the two scientists by name here, Sutton, all right, and Boveri. These two scientists, all right, worked on chromosomal theory of inheritance, all right, wherein they brought a comparison between Mendel's conclusions, all right, and also about the genes and their behavior, all right, especially during the mitosis and meiosis, all right, friends. So but anyway, so these three are not that significant when compared to, and moreover the year here 1900 AD is also important. So Mendelian discovery, rediscovery, in fact, which was through those three scientists, Hugo de Rives, Chemark and Colcorens, is very significant for Genesis. Go for option 3. Then next question we have, dear friends, here. If one parent has blood group A and the other parent has blood group B, the offsprings will have what type of blood group? Which blood group the offsprings will possess? All right. So let me clearly explain this question to all of you. See, first of all, please understand that parent with A blood group, all right, a parent who has A blood group, all right, will have two possible genotypes. One is IA, and one more is IO, all right. And similarly, the parent with B blood group will have two possible genotypes, that is IB, all right, IB and IBIO, all right. So totally now I can say both the parents together, when we consider, they have three alleles, that is IA, IB and IO. All together when we speak, these three alleles are present, which can these three, no, so totally it can make 6 possible genotypes, my dear friends, all right. Please understand that. Okay, what are those 6 genotypes which can be made? See, I will write here IA, all right, then IAIO is the second one, then followed by IB, then we have IAIB, all right, followed by that, all right, then we have IBIO, all right, and finally IOIO, all right. So anyway, so these are the 6 possible genotypes, which can be obtained through, all right, the parents, all right, dear friends. Now let me tell you that, now further, you also understand that, IA and IB are dominant over IO allele, all right. Is also very significant. So the genotypes IA, IO and IBIO will for example, if it is IA IO or IB IO, both will give rise to all right blood groups. That is IA IO will give rise to A blood group, IB IO will give rise to B blood group. All right. So now let me also tell you, due to codominance, all right, where in a heterozygous condition both the alleles are expressed, right, that is what we call codominance. So IA IB alleles, if we consider the genotypes IA and IB, alright, both will be here, alright, expressed. So AB blood group will be produced. AB blood group can be possible, alright. So dear friends, let me tell you that 6 genotypes which are present will give rise to, finally let me tell you, there are 6 genotypes which we have already have explained you. Those 6 genotypes, you can make note of that as well. So those 6 genotypes will give rise to, alright, 4 phenotypes. The 4 phenotypes are what? That is A, alright, B, AB as well as O blood groups. Dear friends, I told you AB is possible because of codominance, and even in the condition IA if it is present, that will give rise to A blood group, or IO is present also, that will give rise to A blood group, because IA is dominant over IO. The similar case is with B. When it is IB IO, it will give rise to B blood group only, or if it is IB definitely IB will be B blood group will be present, expressed. Then when it is IO O blood group can be expressed, my dear friends. So all four blood groups, that is AB and O, are possible, all right, when the two parents, one of the parent has blood group A and one of the parent has blood group B. So you can go for option D, that will be your right answer. Next we have test cross is, all right. First of all, let me tell you what is the definition of test cross here. All right, it is a method for identifying, all right, the genotypes of unknown individual. All right, this is what is the definition of, all right, the test cross is. All right, we can easily understand the genotypes of unknown individual, in fact identify very clearly. All right, so let me perform here on the screen the test cross, how actually it occurs. See, if we consider the F1 hybrid and cross it with the recessive parent, all right, so that will be, the genotypes would be capital T, small t, small t, small t, and the gametes from the same formed after meiosis would be here. These are the gametes, capital T, small t, and here small t is the gamete. So we are going to get now the test cross ratio, phenotypic as well as the genotypic. All right, so capital T, small t, here small t, small t. All right, so 1:1, all right, for both phenotypic ratio as well as genotypic ratio would be 1:1 for the test cross, my dear friends. All right, so basic thing what you need to understand here is test cross, whenever we are speaking, it is capital T small t into small t small t, that is generally F1 hybrid with that of the recessive parent. All right, so next question, let us move. We have gene which is responsible for the synthesis of polypeptide chain is called. All right, see, whenever we speak about this, let me tell you, all right, we need to understand what promoter gene is, what the structural gene is, regulator gene is, as well as the operator gene is. See, first of all, let me tell you, the promoter gene is a DNA segment, all right, that serves to initiate transcription of a particular gene. All right, it initiates the transcription, the promoter initiates the transcription. All right, so it is present upstream, all right, near the transcription start sites. We can find the promoter transcription, where it starts, in those regions we can find the promoter gene, and it is important for starting the transcription and initiating the transcription. All right, further, my dear friends, secondly we have, all right, the structural gene here, right? See, this structural gene is that which codes for either RNA or any other protein, alright? Remember this, which do not serve as regulatory protein, alright? This whenever it codes for the certain protein or alright RNA, that product do not code for regulatory protein, or it does not act as regulatory protein. Remember that. Next time I'm going to tell you what is regulatory protein anyway. Alright. So for example, okay, let me say when we speak about the lac operon concept, the structural genes are lac Z, alright, lac Y, and as well as lac A, which produces various types of enzymes. For example, lac Z produces beta galactosidase, lac Y produces acetylase, as well as lac K here permease is lac Y and lac K is transacetylase. Like this anyway, various enzymes are being produced, but that should not be the regulatory protein. Alright, whatever it produces, it will not act as regulatory protein. Now let me make you understand what regulatory protein is all about. See, when we are talking about this, let me tell you that whichever gene which regulates the expression of, alright, it should act as, or it should help in the expression of structural genes. Those type of genes, dear friends, are referred to as what, children? The regulatory gene. Alright, so here regulatory gene of course are important here. See, here transcription factors bind, alright, and also it is present in the upstream of the, alright, the transcription site. Alright, anyway, so next we have operator gene. What is operator gene here? See, it is also a DNA sequence, dear friends. Operator gene is also what? A DNA sequence. Alright, it is present between promoter region and the first coding gene. Where it is present? This operator gene is present between the promoter, which helps in initiation of transcription. It is present between the promoter and the first coding gene. Alright, so remember that. Alright, and further let me tell you that the operator region, alright, serves as repressor protein binding site. Alright, this operator to this repressor protein binds. When repressor protein is also an important integral part during the gene expression. Alright, so anyway, so it is a site where repressor protein generally binds here. So anyway, here what we need to understand is that the gene which is responsible for synthesis of polypeptide chain is called as what? Are the enzymes, or generally it should be a polypeptide chain, which is nothing but the structural gene. So you have to go for option 2, that is your right answer. 10th question, we have a character which is expressed in a hybrid is called. Alright, yes, it seems to be a easy question, of course. Alright, see, if we consider law of dominance, alright, dear friends, see here, alright, in heterozygous condition, especially when you consider, that means a hybrid condition. Alright, when we consider a heterozygous condition of organisms, when we consider only one character, alright, will be expressed, only one character will be expressed, alright, whereas the other character, alright, one more character will be unexpressed. Alright, only one character will be expressed, dear friends, that we need to remember here. Alright, the character which is not expressed is called as the recessive character, and the character which is expressed in a hybrid or in a heterozygous condition is called as what? The dominant character. So here the answer is A. Please go for it. Next question we have, in melandrium, what is the function of fourth region of the Y chromosome? Alright, dear friends, see here, let me tell you first of all that melandrium, we can also call it to as melandrium album, that is a full scientific name, or it can also be referred to as Silene latifolia. Alright, dear friends, it is a unisexual plant, dear friends. Alright, unisexual or dioecious plant, the male and the female reproductive structures are separate here. Alright, so it has diploid set of chromosomes, that is 24. Alright, and it of course has X and Y chromosomes in it. Alright, so it shows generally XX XY type of sex determination. Alright, so this is general. Okay, now let me come to the question here, dear friends. What is the question? In melandrium, what is the fourth region of Y chromosome does? You know, X and Y both have one standard segment called as segment 4. Alright, this segment 4, which is present on both X and Y chromosomes, helps in pairing as well as in the disjunction, regular disjunction or separation of, alright, the X and Y chromosomes. Alright, X and Y chromosomes, my dear friends. Alright, so now the other chromosomes have 3 segments. Alright, that is segment 1, segment 2 and segment 3. Segment 1, it suppresses the femaleness. Alright, so what is the meaning here is very simple. So it inhibits the female flower development, the segment 1. Then segment 2, which is present in the other chromosomes. This segment 2, will help in initiation of anther development. It initiating the anther development, my dear friends. Alright, so next further segment 3 is present, so it controls the anther development. Segment 3 controls the anther development. Alright, so anyway, so now what is important here is to understand is that what does the fourth region of Y chromosome does. As I said, you both X and Y has 4 segment which help in pairing and disjunction of X and Y chromosomes. So the right answer here is option C. Please go for it. Then the next question we have here is, which law would have been violated, alright, if Mendel has chosen 8 character in garden P? This is an interesting question. Alright, see here, first of all let me tell you that P plant, when we consider the diploid number of chromosomes which can be present, is 14. Alright, so for the same, alright, Mendel chose 7 characters. Alright, 7 characters, the genes for which are present in 7 different chromosomes. Alright, my dear friends, it is present in 7 different chromosomes. He did not select in a single chromosome, my dear friends. It's very interesting to note this. He selected 7 pairs of contrasting characters rather. So he selected where? In the 7 different chromosomes. Alright, so the presence of different chromosomes, now what happened, allowed genes for 2 triads to assort independently. So independent assortment can be done because each one is separated, present in the separate chromosomes, so they can independently assort themselves, they can independently separate themselves. Alright, so that's what, if he had chosen 8 character, my dear friends, 8 characters if he had chosen, alright, so what would have happened? Genes for any 2 characters would have been present on the same chromosome. Alright, see, if I had chosen 8 characters, the genes for the same would have been present on the same chromosome and it would have made complications, right? See, that is why, and would have exhibited linkage. Possibilities where their linkage would have been exhibited there. Alright, for the same reason, alright, even the law of independent assortment would have been violated. That's why he did not choose the 8 character in garden P, dear friends. So the right answer for the question number 12 you have to go for is principle of independent assortment. See, we already are well familiar with law of dominance, so it explains the appearance of a dominant trait, alright, by heterozygote genotype, and law of segregation tells us about segregation of alleles in of heterozygote genotype during gamete formation, and law of purity of gametes, alright, tells us about the presence of a single allele of a gene in gametes. So anyway, that is there. but the right answer here is what? Principle of independent assortment. So you have to go for option C. Okay, here we have the next question. Probability of 4 sons to a couple is. 4 options have been given. 1 by 4, second option is 1 by 8, third is 1 by 16, and the fourth is 1 by 32. Okay, let me explain you this. Say, dear friends, let me consider here. Say, for example, when we consider couple who are planning a child, for example, there are two possibilities, alright, that they can have, alright, either a son or daughter. Alright, two possibilities are there, either a son or a daughter. Alright, so here I can say that probability having either sex is what? 1 by 2. Alright, the two options, out of that any one is possible, right? So that's why we represent it as what? 1 by 2. Probability of having any one of the sex is 1 by 2. Okay, dear friends, now let me say the probability for each child is independent of each other. Okay, in simple terms, let me explain you this. See, probability of 4 sons to a couple is, generally we can represent like this: 1 by 2 to the power n. Here n, which is present, right? Here n represents number of children. It represents what? Number of children. So if we put this formula easily, we can come to know. Alright, see, that means 1 by 2 to the power n means what? 4 sons. Okay, so 4. Alright, so probability if you want. Okay, so that will be 1 by 2 into 1 by 2 into 1 by 2 into 1 by 2, that will be is equal to 1 by 16. So the probability of 4 sons to a couple is what? 1 by 16, dear friend. So remember the formula here: 1 by 2 to the power n. Alright, so anyway we have to go for option, alright, C, that is 1 by 16. Next we have, who rediscovered the results of Mendel's experiments? Here 4 options have been given. Already this we have discussed, my dear friends. Alright, see, I had told you that in the year 1900, alright, 3 scientists, Hugo derives from Holland, alright, then Chemark from Austria, alright, and Karl Correns, alright, from Germany. These 3 scientists rediscovered the Mendel's work, alright, independently, and that paved the way for today what we call it as modern genetics. Alright, genetics has advanced today to such extent that these works, alright, are noteworthy to remember. Because of these rediscovery, only today genetics is to this extent. Alright, and further, dear friends, so these 3 you have to match. So among the given following, the option A itself is correct. But here in the option B, they have given Morgan, they have included here. So Morgan in fact has done a lot of work on linkage. He is called as the father of experimental genetics. How for example 2 genes are linked in a particular loci of a chromosome, that was very clearly explained by Morgan. Alright, and friends, next third option they have included again Morgan. Alright, and the fourth Bateson and Punnett. See, Bateson and Punnett together with alright, one more, Edith Saunders. Alright, see, Bateson, alright, Punnett, and one more scientist called Saunders. Alright, these 3 scientists together, alright, they co-discovered what we call it as the genetic linkage. In fact, alright, later the work was done on linkage by T.H. Morgan, and later he was named as the father of experimental genetics as well. Alright, anyway, Morgan has worked extensively on Drosophila melanogaster to study the linkage. Alright, and very clear results have been given. Alright, to understand the concept called linkage by Morgan. Anyway, my friends, the right option here is that the rediscovery of Mendel's work was done by Derives, alright, Chemark and Karl Correns. So go for option A, that is your right answer. Next we have, in polygenic inheritance triad, which is controlled by 3 pairs of genes. Okay, yes, of course. See, 3 pairs of genes. Say for example, A, capital A, small a, capital B, small b, capital C, small c. Fine, 3 pairs of alleles. Two individuals which are heterozygous for these 3 alleles cross with each other. Okay, the same heterozygous condition, all right, having individual is crossed with this individual. All right, that means capital A, small a, capital B, small b, capital C, small c. Such type of cross, all right, produce phenotypic ratio. In what ratio phenotypic characters are seen, we need to identify, dear friends. When these both are crossed, see, there are what I can say possibilities of here 8 gametes from all this. Okay, say capital A, capital B, 8 different types. Okay, capital A, small b, capital A, small c, capital A, capital C, capital A, small c, then small a, capital B, small a, small b, small a, capital C, small a, small c. The same goes here as well. Alright, capital A, capital B, capital A, small b, capital A, capital C, capital A, small c, small a, capital B, small a, small b, small a, capital C, and small a, small c. So these are the number of gametes which can be obtained, my dear friends. Alright. Now, a cross between these two individuals which are heterozygous for 3 alleles will produce phenotypic ratio. Alright, for there are totally 64 combinations which is possible here. In that, alright, option D is correct. See, 1 will be all dominant allele, 6 will be at least 1 recessive allele, alright, 15 would be at least 2 recessive alleles, 20 would be right 3 recessive alleles, 15 would be 4 recessive alleles, and the 6 would be 5 recessive alleles, and 1 would be alright all recessive allele. You can definitely cross and check out. Alright, see that already gametes which I have shown you, you have to cross. So you are going to get 64 total combinations. Alright, total phenotypes. Out of that, the ratio which we have to consider, as I said you, alright, you have to go for option D. Next question we have, which cross yields red, white and pink flowers, variety of dog flower. Alright, see they have given red, white and pink flowers, right, dear friends. So here let me explain you that. This is explaining the incomplete dominance where the F1 hybrid plants will have different phenotype. Alright, the phenotype is different with respect to the incomplete dominance, my dear friends. Alright, so that means it will have, alright, pink flowers. So the parents generally, if they are, alright, the red and the white, alright, pure homozygous, alright, red and pure homozygous white, alright, the F1 hybrid will be, alright, different from the parent producing pink color. Alright, so this is explaining incomplete dominance or partial dominance or blended inheritance. Alright, my friends, see, let me explain you how it is. Say, if we consider the F1 parents, alright, so capital R, that is red, alright, with genotype I am writing, small r is the genotype, and it is white colored. This is homozygous, this is as well as homozygous. This is homozygous dominant, this is homozygous recessive. Now F1, alright, individual here would be what? Here gametes after production, it would be F1 individual would be what? Capital R, small r, that is nothing but it is pink in color. Alright, when we self this capital R, alright, small r into capital R, small r, in the F2 generation, alright, we are going to get 1:2:1 ratio for both phenotypic as well as genotypic right characters. That means to say, one will be red, two will be pink, and one more will be white. Alright, so anyway, the question as is here, what, which cross yields red, white and pink flower? That means capital R small r into capital R small r. Alright, so we have to go for option C. So here you can just change capital R small r into capital R small r. Alright, so option C will be our right answer. Then dear friends, the next question we have here is, what is the genotypic and phenotypic ratio of, alright, monohybrid test cross. All right. See, what is monohybrid test cross? Already I have explained. It is a cross involved between the F1 hybrid with which is what I can say with unknown genotype with that of its recessive parent, right? So when it is done, what happens? The parent would be here capital T small t into small t. So what happens here? Gametes would be again capital T small t and here small t. So we are going to get here, okay. So let me write the combination here. See here capital T small t, all right, here small t, okay. So we are going to get capital T small t small t small t, all right. So anyway, so now what is the ratio here? 1:1. Both phenotypic ratio as well as the genotypic ratio would be 1:1. All right, my friends. So you have to go for option 1. That will be your right answer. Then dear friends, next question we have. Pleiotropic gene have multiple phenotypic effect. All right. Then multiple alleles exhibit same phenotypic expression, and the last option is polygene exhibit continuous variation. All right. Here again it's quite an interesting question, my dear friends. See, here we need to understand that here, see the statement 1 what they have given. Pleiotropic gene have multiple phenotypic effect. Pleiotropy means a single gene producing many different types of characters. For example, we can consider a disease phenylketonuria. It is affecting only one gene, chromosome 12, but it is affecting multiple organs, many phenotypic characters it is affecting. So anyway, the given statement A is correct, my dear friends. All right. B, what they have mentioned here. See, B they have mentioned as multiple alleles exhibit same phenotypic expression. It is no, see, whenever we are talking about multiple allelism, all right, they of course, you know, when alternate forms of genes, you know, generally will be, you know, expressed, they are giving rise to different phenotypic expressions, my dear friends. All right. Say for example, blood grouping you can see, all right. So the different phenotypic expressions can be seen, all right. So here B statement is wrong. Third statement is what? Polygene exhibit continuous variation. Yes, all right. See, polygenic inheritance is a continuous, all right, inheritance. It gives rise to continuous variations, my dear friends. So without any hesitation we can say that option C is also, you know, correct. All right. So we can say that here among the given options you have to go for B, that is statement A and C are correct. B is incorrect. The next question we have here is sickle cell trait HbA-HbS, which is represented as such, is an example of codominance. All right. It is the assertion, and the given reason is sickle cell trait individual are resistant to malaria falciparum. All right. So one of the dangerous malaria which is caused by Plasmodium falciparum, you know, whoever has sickle cell anemia, they are resistant to it, is the reason which is given here. Now let me explain you this question, my dear friends. Now let me first talk about the assertion HbA and HbS. All right. So these are generally referred to as what? The parental alleles. All right. Both are expressed in the F1 progeny. Yes, both are expressed. Codominance means what? In a heterozygous condition, both the alleles, if they are expressed equally, then we call that as codominance. And yes, of course, sickle cell traits HbA and HbS both, you know, are expressed. So it's an example of codominance. That's the correct assertion which is mentioned here. All right. See, in human beings, if we consider the parental alleles HbA and HbS are present in the form of pair, always they are paired. All right. So sickle cell anemia appears in the progeny, my dear friends. All right. They should be in pair, only then the sickle cell anemia appears. That means you say what? They should be codominant. All right. The given assertion is as such, okay, and it is correct. And let me tell you what is the reason given for that assertion. They have given what? sickle cell trait individual are resistant against malaria falciparum. Alright, yes, indeed, they are resistant. See, several studies have suggested us that in one or the other way, the sickle, hemoglobin, alright, might get in the way of plasmodium. It does not, leave the plasmodium parasite inside our body to infect the RBC cells. So, hemoglobin, alright, sickle hemoglobin, that means in the sickle cells which is present, the sickle cell which are affected cells of sickle anemia, that hemoglobin will avoid, all right, the parasite. That is, which parasite we are talking about here? Whichever parasite, okay, the malarial parasite, all right, malarial parasite, my dear friends. Now here, see, let me tell you, of course, the hemoglobin, it is said, the research says that it will reduce the number of parasite, so conferring some protection against malaria, but not completely. Anyway, here we can say reason is correct, but this reason is not the all right, the answer for the assertion, all right, the reason for the assertion, all right. So I can say that you have to go for here, all right, option, all right, B, that is assertion. If both are assertion reason are true, but both assertion reason are true, but the reason is not the correct explanation of assertion. As I said you, all right, both are correct, but the reason is not the correct explanation of assertion, all right. So that's why you have to go for option B, my dear friends. 20th question we have capital A, capital A, capital B, capital small b, capital C and small c genotype forms how many types of gametes? Dear friends, see, let me tell you, here we use a formula called 2 to the power of n to find out, all right, the number of types, how many types of gametes are present in a particular, for example, polygenic inheritance genotype, all right, 2 to the power of n. So here n indicates the number of heterozygous alleles present in genotype. Say, okay, first let us say we have capital A, capital A, capital B, small b, all right, capital C, small c. This is the genotype, right? Now we'll produce 4 types of in fact gametes. See, let me tell you, you all have all the combinations. Capital A, see, this is the one, capital A, capital B, capital C, capital A, small b, small c, the second one, capital A, small b, capital C, the third one, capital A, capital B, small c. These 4 gametes can be produced. So you have to look for all the combinations. Instead of that, directly you can go for 2 to the power of n, where n indicates what? As I said you, n indicates the number of heterozygote, all right, alleles. Remember that, present in the genotype. Okay, if you go by that 2 to the power of n, say how many heterozygote alleles are present here, right? Say a small b and small c. So 2 to the power of 2, that is 2 into 2, 2 to the 4. Directly you can arrive at a conclusion here, all right. So anyway, dear friends, so here the right answer is what? 4. You have to go for option A. Next question we have, in a dihybrid cross between capital A, capital A, capital B and small a, small a, small b, the ratio of AA, BB, AA, capital B, small b, small a, small b, sorry, small a, capital B, small b and small a, small b, small b in F2 generation is a question, all right. So we need to identify the ratio here, okay. So it is a dihybrid cross, dear friends. See, we already know the parents would be capital A, capital B here, which have been mentioned here, all right. And one more parent is, one is homozygous dominant, one more is homozygous recessive. So the gametes would be AB here from here and AB from here, and when both they fuse, F1 generation would be capital A, capital B, small b. And dear students, when this is self cross, that means to say, capital A, capital B, small b into capital A, capital B, small b, all right. So the gametes which you are going to get is capital A, capital B, capital A, small b, Alright, small a, capital B, and small a, small b. The same goes for the other parent, that is capital A, capital B, capital A, small b, small a, capital B, small a, small b. Alright, when both of these are self crossed, alright, so what ratio we are going to get for this 4, we need to identify, my dear friends. Alright, so here see, okay, let me just put across a b. Alright, here capital A, small b, small a, capital B, small a, small b. Alright, alright, so we are going to cross with the same capital A, capital B, capital A, small b, small a, capital B, small a, small b. Alright, my dear friends, so here see, we are going for the pedigree chart, my dear friends. Alright, so when we go for this pedigree chart, let me say here it is capital A, capital A, capital B, capital B1. Alright, and then see capital A, capital B, small b. Here it would be capital A, small a, capital B, capital B. Here capital A, alright, small a, capital B, small b. Right, in a similar manner, capital A, capital B, small b. Here capital A, small b, small b. Capital A, small a, capital B, small b. Here capital A, small b, small b. Capital A, small a, capital B, capital B. Here it would be capital A, small a, capital B, small b. Alright, small a, capital B, capital B. Here small a, capital B, small b. Alright, here it would be capital A, alright, small a, capital B, small b, and later capital A, small b, small b, then small a, capital B, small b, small a, small b, small b. Alright, so this is what the dihybrid ratio is. Now how much of ratio does this 4 hold? We need to identify, dear friends, in F2 generation. This is F2, alright, generation. Now you observe capital A, capital B, 1. Alright, we have, alright, 1. Alright, then next is what? Capital A, capital B, small b. Here 1 we have, alright, followed by the another. So 2 we have, so 1 is 2, 2 is 2. Then small a, capital B, small b. Here 1 we have, as well as, alright, here 1 we have. So this is also 2, dear friends, is 2. And small a, small b, 1 we have. So 1 is 2, 2 is 2, 2 is 2, 1 is the correct answer. Please go for option C. Then, dear friends, the next question we have here is, Mendel does not select which character in his experiment. Alright, see, dear friends, here, so of course it's a easy question. Alright, see, Mendel chose Pisum sativum for his experiment, that is what we call the pea plant. Alright, so this under in he selected of this pea plant 7 important contrasting characters, that is he selected flower color, alright, then he selected flower position, alright, my friends, then he selected stem length, okay, then followed by the seed shape, alright, then seed color, alright, followed by pod shape, alright, and then pod color. So you can match with these 7 contrasting characters what I have written here. Alright, the character which does not have here is that of plant color. He did not select plant color, my dear friends, so that is the right answer. Next question, dear friends, we have here is genotype is. Alright, see, genotype, whenever we talk of genotype, it is the complete set of genetic material. It is a complete set of genetic material, dear friends. Complete set of genetic material. Very important to note down. Alright. Alright. So it's a complete set of genetic material. In other way, see, genotype can also be used. However, generally or often it is used to refer a single gene. Many a times we used to refer a single gene, or even we use this genotype for referring set of genes as well. Say when Mendelian like checkerboard or experiments we take into consideration, we do it as such. All right, anyways, for example, genotype of eye color, all right, for example, I just said genotype of tall plant, like that, for a particular getting a point character we can select. Anyway, dear friends, or to refer to a single gene or set of genes we use the word genotype, but actually the genotype is what complete set of genetic material or genetic constitution of organism would be the more appropriate word. Among the given options, C, you observe D, that is genetic composition of an individual, that is all right, it is more matching, because you see the other options, genetic composition of many organisms, it is not many organisms, only of an individual. Plastid means we have the chloroplast, leucoplast and chromoplast, it is not the one, all right, it is not the genetic composition of plastids or either the germ cells, it should be of an individual as a whole, all right. So the right answer is option D, my dear friends. Next question is, human egg has, all right, one Y chromosome, one X chromosome, two Y chromosome, one X chromosome and Y chromosome are, all right, the given options. Here, dear friends, let me tell you that, see, when we talk about the human body cell, all right, so it has 46 chromosomes, all right, it has 46 chromosomes, all right. Whereas whenever we talk about, all right, the gametes, especially now it is mentioned here of human egg, all right, it's a reproductive cell or it's a sex cell, all right. So half the number of chromosomes as a body cell is present, that means you reduce it by half, so only 23 chromosomes are present, all right. And when 23 chromosomes are present, already we know allosomes are, out of 46 allosomes, all right, will be, all right, 44, and the allosomes of the sex chromosomes would be 2 here, all right. So now whenever we talk about this, say now out of this, all right, 22 will be autosomes, 1 will be allosome present in the egg. So if you go by that, now let me tell you that, egg cell, has, for example, now human female body cell, when we consider, right, they have X and X as the sex chromosome, so one of the X chromosome will go to the egg, right. So what I mean to say, they will have only one X chromosome, all right, my friends. So the right answer is what? Option B. The next question we have is, how many plants are derived in F2 generation of dihybrid cross? F2 generation of dihybrid cross. Already we know, dear friend, that dihybrid cross means it's a cross involved between the two parents differing in two pairs of contrasting characters. Anyway, so in the F2 generation, how many plants are derived? That's the question here. So let me say, for example, parents which we are selecting are capital R, capital R, capital Y, and small r, small y. These parents will result in the F1 offspring, which will be, all right, carrying, all right, which will be heterozygous for both capital R as well as capital Y. Say that condition will represent it to as capital R, capital Y, small y, all right. So this will yield now, all right, 4 dihybrid plants. It will yield how many plants? 4 dihybrid plants it will yield, dear friends. In F2 generation, it will yield 4 dihybrid plants, and remember, these 4 dihybrid plants will be in the ratio of 9:3:3:1, all right. So the right answer here is 4 plants. In the F2 generation, the plants which are derived are only 4, but in the ratio of what? Phenotypic ratio of 9:3:3:1, all right. Okay, fine. Next question we have here is, the genetic information is carried by, it's rather, a simple question. See here, 4 options have been given: chromosomes, nucleic acid, gamete or cell, cytoplasm. See, the more appropriate answer we have to go here. We know that chromosomes are the vehicles of heredity which carry genes in them, all right. So always the genetic information as a whole, chromosomal theory of inheritance, you can see the genetic information as a whole is carried by the chromosomes, all right. So among here the given 4 options, the right answer should be the chromosome. Please go for option, all right, A. Then, dear friends, next let us move to Next question. Types of genotypes in F2 generation of dihybrid cross. Types of genotypes in F2 generation of dihybrid cross. See, this is of course an interesting question. Let me tell you that. Remember, in F2 generation we are going to get 4 phenotypes, my dear friends. How many? 4 phenotypes. That is, already we know round yellow, alright. Then round green is the second one. Then wrinkled yellow is the third one, and wrinkled green is the fourth one. These 4 combinations we are going to get in the F2 generation, my dear friends. That is phenotypes. When we talk about the genetic constitution, alright, that is the genotypes, we are going to get 9. That is capital R, capital R, capital Y, capital Y, capital R, small r, capital Y, capital Y, capital R, capital R, capital Y, small y, then capital R, small r, capital Y, small y, capital R, small y, small y, capital R, small y, small y, right? Small r, capital Y, then small r, capital Y, small y, small r, small y, small y. You can just observe that there are total 9 genotypes and totally 4 genotypes in the F2 generation of the dihybrid cross, my dear friends. So, among the given options, the right answer you have to go for. The answer is what here? Types of genotypes, right? So totally 9 genotypes have been obtained in the F2 generation of dihybrid cross. So you have to go for option D. The next question we have here is, genetic map is the one that establishes sites of the genes on a chromosomes, establishes the various stage in gene evolution, shows the stage during the cell division, shows the distribution of various species in a region. Alright. First of all, let me tell you that genetic map is concerned with principles of inheritance, alright, and variation. So, see this, it is not concerned with any various stages in gene evolution or in the cell division or in various species in a region, my dear friends. Remember that the gene map or the genetic map, what we refer it to as, it is used to measure, alright, it is used to measure the distance between, alright my friends, the two genes, distance between the two genes. The unit measurement here is done through, alright, map unit, alright. So anyway, my dear friend, the right answer, as I said you, alright, it does not have any role in stages of gene evolution or in the cell division or various species in a region. We don't talk about genetic map there. It is the measure of distance between the two genes, alright, on a particular loci of a chromosome, alright. This is very important to know how far the genes are, two genes for example are present, alright, in a chromosome. Anyway, so the right answer you have to go for here is option A, that is establishes sites of genes on a chromosome. Next we have a child of O group. See, the child has what group? O group, my dear friends, alright. But here his father's group is what? B group, alright. Blood group I am speaking about. So the genotype of father will be, alright. If the child has O group and father has B group, what would be the genotype of father? So very interesting question. See let me tell you that first of all, if the child is O blood group, the genotype of the child would be IO, which is recessive, alright. Then see, as the father is now B blood group, he should have IB, alright, and the other should be IO. This is the only possibility. If this is the case, only then the child can have the O blood group, my dear friends, if the father is B, alright. The only option which you can go for here is option D. That will be your right answer. The last question of today's session is, which of the following conditions represent a case of codominant genes? Alright, we need to consider the conditions for codominant genes, dear friends, alright. See, Alright, it's where again let me tell you, dear friends. So we know already what is codominance. Codominance is what? See, the two different alleles expressing themselves equally in a heterozygote condition is what we call it to as what, my friends? The codominance, right, friends? Say for example, we have AB blood group, right? See, so these both alleles for A blood group as well as B blood group should be expressed in a heterozygote condition. Only then we can say, alright, that too as codominant condition, my dear friends. Alright, now anyway, see, whenever we talk about codominance, it is a phenomenon. Alright, it is a always phenomenon where both genes are expressed equally in heterozygote conditions, but they are expressed independently. That's also very interesting. Alright, they are expressed independently. For example, okay, let me make you clear with the example. If you consider the roan coat color of, alright, cattle, alright, you can observe that there is two different colors on the skin. Two different colors, alright, can be present on the skin. It is very clear now from the fact that that both are of course expressed equally in heterozygote condition but independently. That's very important, my dear friends. So among the given options, what is the first option they are given? A gene expresses itself suppressing the phenotypic effect of its alleles. There is no suppression of phenotypic alleles here. The second, series that are similar in phenotypic effect when present separately but when together interact to produce a different trait. No. They always produce, as I said you, in heterozygous condition both are alright expressing themselves equally. Then third option is allele both of which interact to produce a trait which may resemble either of the parental type. Alright, as I said you, they don't interact, they're independent, my friends. Alright, they're independent but express themselves equally. That's very important. Alright, so the right option is D only, because alleles each of which produces independent effect on heterozygous condition. That is the more appropriate answer which you can go for, my dear friends. So the right answer is option D. Thank you for your patient listening. Please do follow the classes regularly. Alright, so thank you very much.